These protocols are significant because they provide detailed steps for validation of the potency and selectivity of novel HAT inhibitors, which are important research tools and potential therapeutics. The techniques demonstrated in this video are easy to perform and provide information about the effects of HAT inhibitors on global and regional histone acetylation. They make it possible to understand epigenetic regulation of gene expression. Attention to the details is critical. It is important to follow the protocols step by step.
Begin by preparing the enzymatic reaction in a 10 microliter volume inside a 0.2 milliliter PCR tube according to manuscript directions. Then incubate the complete reaction mixture at 30 degrees Celsius for 1 hour in a PCR thermal cycler. Meanwhile, add 2 mercaptoethanol at a 1:10 ratio to the 6x SDS sample buffer. Remove samples from the PCR thermal cycler and add 2 microliters of the prepared SDS sample buffer to each reaction mix. Heat the samples to 95 degrees Celsius for 5 minutes on a heat block. Then cool them on ice. Store the samples at -20 or -80 degrees Celsius, or proceed with gel electrophoresis and immunoblotting. Seed 100,000 MCF7 cells in 1 milliliter of cell culture medium into each well of a 12-well plate and allow the cells to grow to 80 to 90% confluency. When the cells reach the desired confluency, aspirate the cell culture medium from wells 4